Okay, thank you very much for the invitation to Bonnie and Russell, and congratulations to David on his growing medal collection. So, I'm going to try to persuade you in this talk that not only is our schistosomes a cause of a devastating tropical disease, they're actually extraordinarily tractable, user-friendly, and powerful systems for genetic analysis in the lab. So let me sort of run through why that is. So as has been mentioned before, we keep the whole life cycle in the lab. That's unusual for human parasites. Like Drosophila, these parasites produce hundreds or thousands of offspring, so we can do genetic crosses and have very statistically powerful study designs. Then, actually better than Drosophila, these parasites have an amazing characteristic. When a Myrissidia burrows into a snail, it undergoes clonal reproduction. So out of that snail come thousands of genetically identical larvae. So with that natural cloning step in the life cycle, we can actually measure phenotypes with enormous precision, which is incredibly helpful for genetic analysis. So added to that, we now have quite respectable genomes for schistosomes, particularly for Schistosoma mannsoni. We have a growing molecular toolkit. We can knock genes out. We can do transfection. And because of 50 years of experimental and field work, we actually know a great deal about schistosome ecology. And there is a growing list of interesting genetically determined traits that are available to study. These include drug resistance and host specificity and multiple other life history traits. So to sort of illustrate my point, I'm going to tell you sort of three stories about genetic analyses of life history traits of schistosomes. And I should point out, I am the spokesman. The work here has been done by two really driven and talented colleagues, so Frederick Chevalier and Winke Leclèche, ably helped by Marina McDew-White. So the first trait I'm going to talk about is a chronobiology trait. So schistosomes vary in the time that they shed cercari, that cercari larvae are shed from infected snails. This has actually been documented multiple times in different regions of the world. And this example particularly caught my eye, and it's work by Gabriel Mouahid and Hélène Monet, who identified that there are two populations of schistosomes in Oman. We have parasites that shed cercari during the day and seem to be aimed at infecting humans. And we also have parasites that shed cercari in the night and seem to be aimed at infecting rats. And they've established populations of these parasites in the lab in Perpignan. So we want to ask a very simple question. What is the genetic basis for this, what seems a very clearly adaptive trait? So, to do genetic analysis with Drosophila or any other organism, a classic approach is to do genetic crosses and linkage mapping. We can very easily establish genetic crosses in schistosomes. We can infect a snail with a single Myrissidia. We can generate a population of male parasites. We can do the same for a female. We can put these together in a hamster. They mate, produce F1s. We can do the same thing, and we can generate large numbers of F2 progeny. So we've done reciprocal genetic crosses between the night and day shedding parasites. We have then, the people in Perpignan have painstakingly measured shedding profiles of these individual parents, F1s, and progeny. We've then taken parents, F1s, progeny, and genotyped them across the genome. And then we've used a classic approach, linkage mapping. Linkage mapping is 100 years old now, but here we are doing it in schistosomes. So what do we see? There are the profiles that we see in the parental parasites. and very clear patterns. When we look at the F1s, we can see the peak in shedding time has shifted 2 hours to the right, and there's also a small peak in the night. So we're not quite sure whether this is a simple codominant trait or whether we have perhaps 2 or 3 genes involved, but it appears to be a simple kind of genetic basis. What about when we go to the F2s? So I want to emphasize here, this is an enormous amount of work to collect these phenotypes. So Hélène and Gabrielle, to measure these phenotypes, you put a snail in a little jar, and you move it every hour to a new jar, and you count all the cercari that are shed. And they have done it for multiple parental lines infected with the parental strains, with multiple parasite lines infected with the F1s, and for the first cross, 114 independent F2 genotypes they looked at. And they didn't look at them once, they did many of these parasites over multiple nights to check that we actually have repeatable traits. So incredible amount of work. And what do we see when we look at F2 phenotypes? Well, we don't see quite such simple patterns, but we see enormous heterogeneity in the patterns that we see. So on the top left, you can see a shedding pattern for one particular F2. It's a night shedding parasite, like one of the parents. We have just below it a day shedding parasite. And then we have a whole load of profiles that are somewhat more complicated with multiple peaks or intermediate peaks. So we scratched our head for a little to think, how are we going to categorize these traits? And in the end, we came up with a very simple metric. We simply measured the proportion of cercari that were shed during the day. And sure enough, when we look at snails that we have shed on 2 different nights, this is a beautifully repeatable trait, with, so we know we have a strong phenotype we can use for our genetic mapping. So we now take all of those parasites and we want to genotype them. So we don't want to sequence whole genomes, that will break the bank. So we have just partially sequenced genomes by exome capture and sequencing. So we essentially, we make the 363 megabase genome into a 15 megabase exome, and we can therefore sequence much cheaper to high read depth. We can specifically target only genome regions that can be unambiguously aligned, and we can get it down to around $200 per genome. So our exome capture covers about 92% of the exome, and here's a sort of picture of the kind of coverage we get across the 7 autosomes and sex chromosomes. Here we've sequenced to about 300x depth. This is actually a female parasite. You can actually see on the Z chromosome half the read depth in the Z-length region. So we have a very expensive sexing method here. Anyway, it works very well. So now we do that for our sort of 100 or so, or I think 65 progeny for each of these crosses. So we've now, this is the workhorse in our lab. We've now done over 1,000 exomes, and the queen of exome sequencing is Marina here. So what do we see? We do a classical linkage mapping, so we essentially screen across the chromosomes, searching for regions where our genetic markers segregate with this shedding trait that we're interested in. And sure enough, we see a very strong peak on chromosome 1. And I should mention, the dotted blue line is a permutation threshold. We randomize our phenotypes against our genotypes, rerun this analysis 1,000 times, and so that tells us where we will get peaks at random. And the peaks we actually observe are much, much higher. We see strong evidence for involvement of chromosome 1. We see some evidence on chromosome 6 as well. So what genes are in those peaks? Well, there's about 160 genes under that peak on chromosome 1. And when we look under the very highest part of that peak, there's a very compelling candidate gene. HES1 is a transcription repressor that influences cell proliferation, embryogenesis, and development in Drosophila work. So a very exciting candidate. We will, in the future, do functional assays to see whether really that is involved or whether we need to look further. We were also interested in the idea that sort of clock genes were involved in this periodicity trait. So we looked for homologs of clock genes, and we find a whole load. None of those are under our QTL peaks. I don't think those are involved. Okay, let's move to a new trait. So Winker was interested in the number of cercari that are shed. This is obviously important for transmission, but it's actually quite variable when we look at lines both in the field and in the lab. So she looked at 2 parasite lines. LE produces thousands of cercari. SM Bray, a Brazilian line, produces miserable numbers of cercari. So we wanted to know, again, what is the genetic basis for this important, ecologically important trait? So, and just to give you an idea of how consistent this trait is, she monitored the parental parasites over 4 weeks, once a week, and you can see shedding for the Brazilian and the LE strain, very consistent, strong differences in phenotype between these parasites. And that's associated with big differences in virulence as well. So looking at the genetic basis here may tell us something about virulence evolution. So, and we know from previous work, from Joanne's lab, from Joanne Webster's lab, this is heritable. She is selected in the lab and showed that you can actually change the phenotype over very few generations. So again, reciprocal genetic crosses, 188 progeny in 2 different crosses, and we had, I think, 4,500 informative SNPs fixed for alternative alleles in our genetic crosses. So what do we see? So here, same kind of plot. We see 3 genome regions showing very strong linkage with cercarial number. Again, we see the middle of chromosome 1 appears to be involved. We see a big peak on chromosome 3, and we see, again, a big peak on chromosome 5. In all cases, highly, highly significant. So we know now we can actually detect polygenic traits using this kind of linkage approach, and we see 3 major QTLs. And we can actually predict, using our genotype data, the phenotype of the progeny. So we can take the genotypes at these 3 QTL loci, and we can see how many alleles from the high shedder parent are found in each of the progeny. And sure enough, there's a beautiful relationship between genetic variation at these 3 QTLs and cercarial shedding. Okay, third phenotype. So this is a classical phenotype, compatibility between snails and parasites. And this is a subject that the group in Perpignan has done an enormous amount of work on. And really, this project was kicked off at an excellent meeting in 2013 in Perpignan. So we wanted to really take this down to a very reductionist sort of level and just take 2 pairs of snails and parasites with different patterns of infection. So LE infects 2 different genotypes of glabrata. BG Bray infects, SM Bray infects its sympatric snail, but it cannot infect BGBS90. So we want to know what genes allow LE to infect BGBS90. And again, it's a simple problem of conducting genetic crosses. But a problem here, if we have a system where we infect snails with Myricidia and nothing comes out the end, we have nothing to genotype. We can't do our standard genetic analysis. So we borrowed stolen a trick from yeast and malaria genetics, which is to use linkage analysis, but with pooled sequencing of progeny. So we divide our F2 progeny into 2 groups. We expose one group to selection, whether it's drugs or in this case, the snails that we use. We then sequence these pools of parasites coming from the two snails, and we see where there is a big distortion in allele frequencies. So let me just talk you through this. So in this cross then, when we put our F2 progeny with all their shuffled genomes through BS90, many of those larvae will die and they won't emerge. BG Bray is permissive, all the parasites go through, and so we have a comparison and we can identify genome regions where we have enrichment of genes from the LE parent. And this is pooled sequencing, so we measure allele frequencies using read depth in our Illumina sequence. So what do we see? So this is the comparison. We're taking pools of Cercari, and it's not just from one snail. This is from 700 snails in each of this group. Really big experiment. And we see very, very clear patterns. So on the y-axis is the minus log p-value. We have values up to 10 to the minus 20. We see a very strong peak on chromosome 3, another peak on the end of chromosome 2, and some possible peaks on chromosome 1 and chromosome 4. So two major alleles appear to be key to explaining host specificity in this particular system. What about if we go to an independent system? So Benjamin Gobal in Perpignan set up another cross. This time he used SM Bray, as we did, but he crossed with Guadalupe parasite, SM Gua. And again, the patterns are very similar. SM Bray in this case can infect both snails. SM Gua can only infect BG Gua. And crosses were done in Perpignan. We did all the sequencing and analysis. And what do we see? We see a stonking great peak on chromosome 3. Again, we have a major locus involved in compatibility on chromosome 3. And we also light up that same region on chromosome 2. So two independent crosses, and we see chromosome 3 in particular lighting up in both of these. So we've got three traits now. We can now actually do a sort of map of these QTLs. We look at compatibility, two major loci. We look at circarial shedding. We hit the same region of chromosome 3. I strongly suspect it'll be the same locus. We look at chronobiology. We hit the same region of chromosome 1 and an additional region on chromosome 6. So we've got sort of key genes that are linked in several traits. And in future work, I hope we will home in and identify precisely what genes and mutations are involved. And summary, we can now actually do great work on really understanding the genetic basis of these ecologically and evolutionarily important traits. Thank you very much. Many people contributed to this.